Now we move to our next speaker, Professor Dominik Obrist. Hello, Dominik. Hi, Dominik. Could you please share your screen and
yes, hello.
Talk. Yeah,
yep. Can you hear me?
Yeah, yes, great.
Okay,
Hi, Dominik. So I will introduce you in Chinese to our audience, okay? And after I finish, you can start with your talk, okay?
Okay.
Dominik Obrist 教授，他是呢应用数学博士和机械工程的硕士，是伯尔尼大学生物医学工程研究中心心血管工程教授，同时呢也是一家瑞士医疗技术初创公司的联合创始人，是医疗技术学术和工业两栖专家。那他的主要经历呢，包括在瑞士苏黎世联邦理工学院建立生物医学流体动力学研究小组，并在美国知名的超级计算机制造公司担任包括研发和市场等等诸多重要职位。那该公司目前是惠普公司的子公司。Okay, Dominik, I'm done with your introduction. Please, the stage is yours.
Thank you, Vivian. So let me share my screen. I hope you can see it now. So I'll be talking just for a second. Yeah. I'll be talking to you about my views and innovation trends 2023 in cardiovascular applications is my field of expertise. So when we're looking at new trends, it's usually a good idea to look a bit back in history and understand where we're coming from. And I put together this picture with what I believe is a bit of a history of the technologization of cardiovascular medicine. It all started surprisingly long time ago, 80 years ago, 1953. We have on the bottom left, we have a picture of the first heart-lung machine. And if you go to the surgical theater nowadays, you'll be surprised that the machine looks almost the same. Not exactly the same, but still very similar. Soon thereafter was the first pacemaker that could be implanted. There was the first artificial heart valve. There was the first ventricular assist device. You see here Drs. DeBakey and Liotta doing the first surgery in 1966. This was a long time ago. And this really were this disruptive technologization of this medicine happened, where a lot of technical devices came on board. And it took a while until the next major innovation came about in 1977. We see here Professor Grünzig in the University Hospital in Zurich, who did the first balloon angioplasty of a coronary vessel, of a coronary artery. So that was the onset of this catheter-based medicine that we see nowadays so often. A bit later on, we saw the stent coming. This is really standard technology with which heart attacks are nowadays treated. So this all happened, an awful long time ago. So where have we gone since then? If we look at more recent trends, I would like to summarize this all a bit under the topic of miniaturization and integration. We're trying to make things smaller, to integrate things more. We're trying to use this catheter-based technology a bit more. So nowadays it's standard to use these vascular grafts to do surgery on the aorta, right? If you have an aneurysm in the aorta, you don't open the chest anymore. You can directly do this via catheter. We have learned how to implant heart valves via catheter without opening the chest. And we have learned how to make pacemakers so small that they can also be inserted via catheter. So these are all phase of miniaturization. Also down here, maybe that one, the Impella ventricular assist device, which can also be inserted by a catheter in acute situation. And maybe to go a bit on a larger scale, here we have the Carmat heart, this total artificial heart, a complete heart that can replace a diseased heart, a failed heart. So all these recent trends went toward miniaturization and integration at the same time. To be a bit provocative, they didn't really invent any completely new idea. They just made existing technologies better and smaller. If we look at more emerging trends we are starting to work on today, and maybe in the next few years, we'll be talking about that more. We look more and more at patient-specific devices in cardiovascular medicine. Certainly, 3D printing plays a big role here. We have here on the top right, a nice project of colleagues at ETH Zurich, which they are printing 3D heart valves. They can be made specific for a patient. They can be given specific mechanical properties, some anisotropic materials, very nice, and you can do a lot more with 3D printing than with just these standard size heart valves that we have nowadays. Ad hoc fabrication, but also what we see down here, a company Celtis in Holland that is currently running clinical trials with in situ tissue engineering. So they have a graft, a porous graft, a scaffold, which they insert. And then while it's already in the body, it works like a bioreactor where tissue grows on it, eventually replacing the scaffold and having a tissue engineered new vessel. So again, this is great new technology, but also this is just making existing things better, more, let's say, from a materials, from a fabrication side. What I would really like to talk about today is a new field, one of my favorite fields, is the field of microvascular diseases. And why do I see this as an emerging trend? Well, microvascular diseases, of course, they have been recognized by medicine, by science for a long time, but in clinic, in the hospital, they have been neglected. Why? Well, because they could not be diagnosed. You do not see it as too small, and they could not be treated. There were no tools around, no technologies to really treat them. So by becoming able to address diseases in the microvasculature, we're not just making all technology better, we're really opening up a whole new field, or in terms of a company, we're opening up a potential new market. The problem is the scale challenge. All the tools that we have in medicine nowadays, pointing here at the catheter, they go to a certain smaller size, maybe millimeter, maybe sub-millimeter scale. But where the lesions are, we're looking at micrometers or tens of micrometers. So there's a scale gap we need to learn how to overcome. How can we use our tools, our things that we're using in the clinic to treat and diagnose these small scales? Microvascular injury of the microvasculature is everywhere. It's connected to a lot of well-known diseases. We see here heart attack and stroke, the two biggest killers in the industrialized world. They are directly connected to microvascular injury, which nowadays is usually untreated. But also dementia, right? An important topic, especially in the near future, is also directly connected to injury of the microvasculature in the brain, and many others. I would like to focus my talk now on heart attack, where we have specifically working on in the last few years in my team. And we're looking specifically at the problem of microvascular obstruction. This is a condition that is occurring in more than 50% of all heart attack patients suffer from microvascular obstruction, even though it's usually not diagnosed. So what is happening? Look at the picture on the top left. We see here a larger coronary artery with a blockage in it. This is the heart attack. This is the thing that blocks the blood flow to the heart. We have very good technology today to treat this. Heart attack is being treated by catheter, with a balloon, and with a stent. This works really well. But what is happening after that, down here in these locations, this is usually not diagnosed and not treated. This is small debris falling off the primary thrombus, the primary obstruction, and being washed down into the small vessels and creating blockages there, right? So we have the big blockage in a large vessel, we resolve that one, but we end up with small blockages in small vessels where the catheter cannot reach anymore. People with MVO have a significantly poorer outcome. The event-free survival drops over several years by many, many percent. So we definitely want to diagnose it and we want to treat MVO. The reason why it's not diagnosed is clear. It's occurring in vessels smaller than 0.2 millimeters. Our imaging technology, especially the one we're using in the acute situation, the catheter lab in the hospital, does not resolve these scales. So they see vessels of 1 millimeter, maybe a bit less, but the small vessels, they don't even see. So this is not being seen, it's undiagnosed. What about treatment? Well, even if you knew that there is an MVO, there's no good treatment. First idea is to do systemic drug administration, do a lysis drug. So you infuse it in the arm, but this doesn't work. Next best thing is you use the catheter and bring the drug directly to the aorta, hoping you bring more drug to the local MVO. But also there, the dilution is too high. So recently people try to insert the catheter directly into the into the coronary circulation as far as they can. But also there, you have a problem that not enough drug arrives at the actual lesion. Most drug takes a bypass at the location where there's no obstruction, right? So the underlying problem, a little bit of physics I need to do here in this talk, is the following. We're looking at reversibility of low Reynolds number flow. Look at the experiment on the left-hand side. We have a syringe with a red dye, and we're infusing this red dye into this reservoir up here. Right? This is a situation at Reynolds 300. This is like what we have in a real artery. So pressing the syringe now, and we're infusing. And it comes in like a jet, and it's nicely mixing with the fluid. As we retract the plunger of the syringe, the drug or the red dye stays in there. This is what we expect intuitively. Now look at what's happened in the microcirculation, where we have a small Reynolds number. This is a typical property of the microcirculation. Same experiment, except for different Reynolds number. We're infusing and we're pulling back. Right? So the flow is completely reversible. We cannot just inject the drug with the syringe and expect that it stays in there. We're suffering from flow reversibility. That's a fundamental property of microcirculation. That's why drug infusion is so inefficient. Our approach is the following one. We're working together with a startup company called CoreFlow Therapeutics. They have developed this console and a multi-lumen catheter. There's a balloon to locally block the coronary artery. There's an infusion opening and an access for a pressure wire. Instead of just placing a catheter into the coronary vessels and infusing the drug, we inflate first the balloon, thereby temporarily disconnecting the vascular bed from the bloodstream, and then infusing the drug. This will give the drug more time to dive into the microcirculation and do its job as it's supposed to. Let me show you a video here. This was done in our lab in an in vitro experiment, but there's also animal trials running, also clinical studies have started now. What you see here is a model, a microfluidic model of a vascular bed. On the left-hand side, we're doing the experiment without the balloon. On the right-hand side, we're inflating the balloon first before infusing the drug. And you see the drug coming in, it's pulsating, it's a heart that's pulsating. The high concentration is denoted by yellow color, low concentration by red and black. After about 10 seconds, the infusion stops, and the drug that has been infused on the left-hand side is already being washed out. It's getting darker and darker, whereas on the right-hand side, where we still have the balloon occlusion, we still have a very high drug concentration. And even far down here, where we have the occlusions, we have a significant level of drug reaching the occlusions. And so this is a much more efficient way of administering the drug. And you see now, after that time, when almost all the drug has washed out on the left-hand side, we're still fully perfused with high concentrations of drug on the right-hand side. So this is this high efficiency that we can reach with this balloon catheter technology. After balloon deflates, of course, there's also washout, but also here you see that the right-hand side, where there's no obstruction, is washed out efficiently, whereas on the left-hand side, where we have no microvascular, where we have the obstruction, the washout is less efficient and the drug stays in. If we quantify this in terms of reach concentrations, we see here, especially in this diagram, that without balloon versus balloon, we can improve the maximum drug concentration by factor 2.2. If we are going for the accumulated drug dosage, so the integral over time, reaching better results, even a factor of 5.2. So I believe diagnosis and treatment of microvascular pathologies is one of the new frontiers in cardiovascular technology. I think we have now a good technology platform for targeted substance delivery. We see this on the right-hand side is the newest version of the company currently used in clinical trials. We can also use it to measure microvascular resistance. I have not talked about this. We can do a targeted drug delivery. This also gives us the opportunity for microdosage, so reducing the dosage of potentially toxic drugs. But we're also thinking beyond that and thinking of, well, we could also use the same thing to administer not drugs, but to transport micro robots like the ones we show down here, micro robots who do local microsurgery. But this is for the future, but I think we're now at an interesting point in time where we're exploring a completely new field of pathologies. So thank you for your attention.
Okay, great. Thank you so much, Dominik, for your very nice presentation and your great contribution. Of course, I have here a general question regarding the impact of AI, artificial intelligence, and machine learning, things like that. That is really a hot topic worldwide. How is artificial intelligence being incorporated into cardiovascular medical devices, and what impact do you expect it to have on patient outcomes?
Right. Yeah. So I think artificial intelligence nowadays is abundant. It's everywhere, and it's not a technology that we choose or not choose. It's just something we're using anyway, and the only thing we can do is to use it the right way, efficiently. What I believe the most efficient use nowadays for artificial intelligence in cardiovascular medicine is in the imaging technology, right? We're looking at segmentations of imaging, of images, which has been done manually, very time consuming, very expensive, and can be done automatically now. We're using this in my research group, but also in a hospital, which is just next door from my office. We're seeing this more and more used nowadays. And this is definitely something which has a big indirect impact on the patient. They will receive the same treatment, but it will cost less, it will be faster, it will be more repeatable for them.
Great. Thanks again, Dominik.